Hello everyone, my name is Fatemeh and today I'm going to be talking about the antimicrobial performance of a proprietary metal oxide sol coated onto polyethylene and oxidized polyethylene against Pseudomonas aeruginosa using confocal microscopy imaging. So with the now prevalent use of plastics and polymers for many applications, due to their moldable and durable nature, there is a growing interest in developing and mass-producing antimicrobial polymers. So metal oxide sols has been the chosen route for my project because it can be easily and cheaply prepared with simple manipulation to coat complex shapes. We use polyethylene as a starting point because it has many uses. As polyethylene is nonpolar and inert material, chemical oxidation was performed at various degrees as to improve sol adhesion. So this was done for 2, 10 and 30 minutes as a starting point. So overall, the aims of my project here looks at preparing a proprietary metal oxide coating. So here we've named it Sol A and Sol C for simplicity in this case, where Sol C is the same oxide, but it's aged to form larger sols, which these are used to coat both the polyethylene and oxidized polyethylene materials. Another aim is to develop a technique to evaluate surface biocidal activity aimed for initial biofilm development, and to assess changes in initial bacterial attachment from the treatment. So moving on to the experimental aspect of this piece of work, to test the sol durability under aqueous environment is important as the assay is conducted in a hydrated setting with microscopy imaging. And to see the extent of leaching is important as well. So inductively coupled mass spectrometry was used to determine the metal concentration in water after sequential washes, and then XPS was used to determine the elemental composition. So moving on to the bacterial assay. In this poster, I presented my work on 2 microscopy assays using these materials. So I've looked at attachment and viability of the biofilm by staining the biofilm on the surface with dyes. So I used Cyto9 alone to stain, to see attachment, which stains both live and dead cells. And I used the viability kit from Invitrogen to look at changes in viability. So this kit has Cyto9 and also propidium iodide, which is another dye. The propidium iodide stains membrane damaged cells and effectively looks at the dead cells on the surface. So the ratio of these dyes shows the degree of change in viability. So moving on to some results. Leaching studies have shown that negligible amounts of metal is leaching off, well below the safety limits for this metal oxide. And the XPS results are shown here, which you can see there are significant modifications that has occurred to the material after treatment, including oxidation treatment and coating of the metal oxide. So here I provided some images, example images, AFM images to visualize the particles. What we can see here is 2 examples of both roughness and stiffness parameters of both the sols, where aging has shown to be successful, forming Sol C from Sol A during the synthesis stage, as the particle size has increased about 10 nanometers and forms quite globular structures in general. So moving on to the bacterial assay results, the microscopy assay provides images, which I have analyzed using ComStat. This is a plugin for ImageJ to analyze the pixels into percentage area occupied after thresholding. What we can see is that the increase in oxidation pre-treatment times shows reduction in attachment, particularly for Sol C. And I provided some example images where you can see that there's practically very little or no bacteria in this image compared to the control. This is important as attachment is an important step to biofilm formation, and to prevent attachment is an advantage in preparing an antimicrobial material. So looking at the viability assay, where we've used both the dyes, there is a significant reduction in the percentage area occupied in the ratio of live and dead dyes used upon surface modification, particularly with Sol C again. I think further study is needed to understand the scope of the material. Currently, I am focusing on the antimicrobial effects of the oxidation treatment alone, as this in itself shows promising results in reducing attachment and viability against gram-negative bacteria. So I think that's it. Please feel free to ask any questions, and thank you.